relatively new to the Northern Forestry Center. I'm started in June of last year, but I did a lot of work here for the last 10 years at University of Alberta, particularly with Dr. Nadir Erbilgin. So a lot of what I'm going to be presenting is some work that I did with him that you may be interested in about mountain pine beetle, together with some future work that I will be working on. And if you have any questions, you can ask me to stop whenever. So I'll be talking about early detection and management tools for mountain pine beetle in Alberta. So mountain pine beetle, I'm sure in Peace River, everyone knows about it, but I just like to give a little intro. It's a small beetle, only maybe about 5 millimeters long, but it lives underneath the bark of trees for its development. And in that photo is what we call bark beetle galleries, and the little white dots are the pupa. So it's going through its development underneath the bark of trees. So it has this tight connection with trees that I'll go into next. And it's a native bark beetle to Western North America. In the map, you can see the black area is where mountain pine beetle is native to, and that corresponds with a lot of different pine species, including lodgepole pine. And then when you go up into British Columbia and Alberta, lodgepole pine continues north and continues east, and these areas have traditionally or historically been out of range for mountain pine beetle because of how cold it is. However, in 2006, because of warmer winters and a large outbreak in British Columbia, it breached that Rocky Mountain boundary and came into northern Alberta and infested lodgepole pine, lodgepole and jack pine hybrids, and even some jack pine. So I'd like to show this diagram of the life cycle, because it lets me highlight how mountain pine beetle and its host tree, so in this case, lodgepole and jack pine, have this close connection. And it's this connection that we can use to understand, say, where mountain pine beetle is in the landscape, or what trees are suitable or not suitable for mountain pine beetle. So during the dispersal and host selection stage for mountain pine beetle, so this is the adult leaving the tree that it developed in. It's wanting to find a new tree, and females are these pioneer beetles that find the new tree. They're going to use tree chemicals, among other things, but I'm going to focus on chemicals and defenses. So they use tree chemicals to find a new tree. And then during the aggregation and host colonization stage, they can also, both the males and females, produce insect pheromones to attract and also to do anti-aggregation. But essentially, they use chemicals to communicate. And some of these chemicals are actually derived from the tree chemicals, the chemicals that are in the tree, like alpha-pinene. But the trees are also producing defensive and toxic chemicals. And these the beetles have to interact with. Some can be toxic, some the beetles actually use. And beetles also bring with them blue stain fungi. These blue stain fungi help with this aggregation and host colonization stage by essentially detoxifying some of the defense chemicals for mountain pine beetle. And another way that these beetles and trees interact is during the development stage. So during this time, tree chemicals are really important, especially during low population periods where mountain pine beetles at eking along and not outbreaking. So kind of what we are in right now in most of Alberta and BC. These defense chemicals can be toxic to the developing insect, but again, the blue stain fungi can help detoxify them, plus increase the nutrients of that phloem tissue. So there's these multiple ways that insects and trees interact. one of the mechanisms that I look at are these defenses, and that's what I'm going to focus on today, using a couple different experiments and some ongoing work. And the three main topics I'm going to talk about are host suitability assessments. And this is some work that I did with Nadir Erbilgan at U of A, and talk about some future directions in jack pine, because that was looked at, what I looked at before was lodgepole. Next, the main topic I'll talk about is detection. And this is particularly important while they're at these low populations, and some ongoing research that I'm going to be doing with researchers here at the Northern Forestry Center and Pacific Forestry Center. And finally, some kind of off the wall idea, though not that off the wall, because it's widely used in agriculture, but biocontrol and fungus that might be a useful biocontrol, but I'll talk about what's been done and what needs to be done for this to be a potential control management tool for mountain pine beetle. So first, I'm going to talk about this host suitability, and I'm going to go back to what that life cycle showed, was that chemical defenses and defenses in general are this way that trees and beetles interact. And I'm going to go into some detail. So it's not just all chemicals. All chemicals are not created equal. So it depends on the composition of these defense chemicals, essentially the chemicals that make pine smell like pine, and their concentration that could be toxic to mountain pine beetle. There is some there is literature that shows that limonene, a monoterpene that's pretty high in concentration in lodgepole pine, is toxic to mountain pine beetle and its associated fungi. So I'm kind of looking for what are these chemical signatures that might indicate a suitable host versus not suitable. And another form of defenses trees have are resin ducts. And that little photo shows a line of resin ducts. And these are filled with chemicals, but we call them anatomical because they're essentially barriers to a mountain pine beetle, or the fungi, in particular the fungi, from invading into the wood. And research has shown here in Alberta that a larger number and size of resin ducts can be associated with resistant trees resistant to mountain pine beetle. And again, that's lodgepole pine. So to start teasing this apart, to try to find what are the essentially biomarkers for a suitable tree versus a not suitable, I was part of a project called Res4 at University of Alberta with Nadir Erbilgan and Barb Thomas, where we used provincial progeny trials, where they know the parentage of the trees growing in a plot, essentially. And these were 35-year-old trees, and we phenotyped them in that we quantified their defenses. And then a grad student named Aziz Ola in Nadir Erbilgan's lab, he conducted some insect and fungal bioassays to essentially tease apart what chemicals are more toxic, but importantly, not just a single chemical, but what group of chemicals are more toxic to a mountain pine beetle, as a way to essentially screen trees for suitability. And he did this in the lab using a great technique, mixing phloem and wood of trees with agar and some nutrients, and seeing how the beetles grew or accepted this media when adding different profiles of chemicals. And these profiles of chemicals came from the families of a lodgepole pine that we phenotyped. So we went out there, we knew the parents of these trees, and we measured their chemical defenses in that we measured the quantity and quality of monoterpenes, and that's what's on the essentially y-axis of this heat map. And we can see in this heat map that so light indicates a low concentration and black box indicates high concentration, and that these different families that are these arbitrary numbers on the x-axis have different profiles of these chemicals. So some have high proportion, say alpha pinene, but low camphene, and vice versa. So there's this variability in the profiles of these trees that may be defensive to mountain pine beetle and be more or less suitable. And through his bioassays, he found that, yes, these different profiles showed different responses by mountain pine beetle. So this is weight change, which is a good indicator of how successful that beetle will be in producing eggs, because a heavier beetle can produce more eggs and is more successful in being able to get through that tree phloem to lay its eggs. So he found that some families, like 1799 on the x-axis, had very low weight change, actually reduced in weight, so did not accept the media. So those chemicals in that family were essentially toxic and not suitable to mountain pine beetle, whereas families such as 1046 and anything that has an A on it, those were no different from controls. So those were very suitable to mountain pine beetle. So those would be families that if you were screening for suitability for resistance to mountain pine beetle, then you wouldn't choose these families necessarily. He also looked at a very important chemical in lodgepole pine called beta-phellandrine. So we had to leave these out of the bioassays because it's a very expensive chemical, but it's very high in concentration. Nearly 50% of the entire monoterpene profile is this chemical. And so we chose some low, medium, and high percentiles of this based on the variability of these progeny trial families. And he found that higher amount of beta-phellandrine was less suitable to mountain pine beetle. And this is important. I kind of leave this slide in here because I want to point out that lodgepole pine has beta-phellandrine, but jack pine does not. And beta-phellandrine is pretty toxic to mountain pine beetle, but jack pine is missing. Pure jack pine does not have any beta-phellandrine in it. So that might be something that needs to get looked at in jack pine when looking at suitability based on chemicals. So with his results, I essentially wanted to find how can we categorize trees based on suitability from Aziz's bioassays. And so I ran a regression tree and essentially found out that the proportion of limonene was, when it's high, those families tended to show poor performance by mountain pine beetle in the lab. So limonene, not just the concentration, but the proportion of limonene of the total monoterpenes was an important predictor in whether mountain pine beetle would be successful in that lab-based bioassay. And so with that information, we can kind of start ranking our families based on the amount of limonene or the proportion of limonene that it has, the concentration of a very minor chemical called gamma-terpenine that was also came out in that regression tree as predicting success of mountain pine beetle, and beta-phellandrine. So we can start looking at some families are less suitable and some families that the province has in these progeny trials are more suitable. But he also ran some fungal bioassays too, same kind of deal where The fungi, the blue stain fungus that beetles bring with it, was grown on media that we put the different chemical profiles in based on that first heat map slide. And he found that different families showed different growth of Grossmannia clavigera, this blue stain fungi. And again, showing the variability that we're finding out in the trees that the province has in these progeny trials with the chemical signatures and response with the fungi, which again is that really tight connection that beetles have with their tree and fungi. So all of those are needed for a successful infestation. So families like 1867 and 1865, again, these are just arbitrary numbers, but it's what they're called. Those had very poor growth in Grossmannia clavigera, or at least those profiles showed, whereas families like 1046 or 1005 had really good growth, no different than our controls that are on the far left. So now we can add that information to, again, this ranking of tree families. If someone wanted to, say, choose trees that were least suitable or more suitable if they wanted to mountain pine beetle to do further tree breeding with or selection of, we can find that it's not just limonene, but also alpha-pinene, camphene, and a suite of negative, the monoterpenes from the literature that are shown to be negative to mountain pine beetle that can help segregate some of these families that are least suitable from the families that are more suitable. And again, this could be used in a tree breeding program and or identification of potentially suitable trees elsewhere. However, we also have to add resin ducts to this. And in a paper that I talked about earlier, we found that that higher number and size of resin ducts are resistant to mountain pine beetle. So it's kind of a combination of the chemical profiles with trees with a high amount of limonene being least suitable and trees that also have a high number and size of resin ducts also being least suitable, all the way down to if they have low chemical concentrations or low proportion of limonene and low number or size of resin ducts, those would be the most suitable to mountain pine beetle. But these were done all in lodgepole pine, and that all came from kind of the central Alberta area. So the Whitecourt, Swan Hills, maybe a little bit further west. That's where those families came from. And so we should expand this further to look at host suitability based on these chemicals and resin ducts of jack pine. So this is I should have probably put this photo earlier in the presentation, but you can kind of see the close-up of the resin ducts that are in the wood that we can measure using increment coring. And so further tests that I'm going to do are look at the natural variation of resin ducts in lodgepole pine. I want to look at whether site and stand characteristics impact these defenses, and also whether there's some climate sensitivity of defenses, as in when things are dry and there's a drought, mountain pine beetle tends to take advantage of that situation because trees tend to have low defenses in chemical defenses. And how sensitive is jack pine to these times of drought? Do they also lower their defenses? And I can measure that through history using their resin ducts. And this is also a neat technique, using these chemical defenses as essentially a proxy for whether mountain pine beetle would infest those trees, because mountain pine beetle now is at pretty low populations. And so I can't rely on what trees have been attacked and not attacked to be able to do this work, but I can still continue using resin ducts and chemicals. So next, I want to talk about detection, and I'm going to continue on this theme of low populations because a lot of the detection tools that we have for mountain pine beetle rely on high populations. So the chemicals that we've used to put on traps are mostly tested where populations of mountain pine beetle are high. And also our aerial surveys rely on easily detectable red trees in more than just a single one in a sea of forest for you to be able to identify where mountain pine beetle is attacking. So you might want to know what the current status of mountain pine beetle is. So this is a map from, it's actually 2021 when it was last updated, but in orange, you can see the accumulated affected stands since maybe 2006 up to 2020. And you can see it got as far east as just north of Lac La Biche. However, in red, you can see that there is a shrinkage, essentially, of that affected stand. So there's lower area in 2021 that is being impacted. And so there's just kind of this general decline in mountain pine beetle since its peak. And there are some areas with sustained outbreaks, such as west of Calgary and around the Cypress Hills area. But in general, we're seeing lowering of the population. But this gives us opportunities to enact effective management. But the problem is we need information. We need information on what maintains low populations and how to find those low populations, and also how to measure when populations are increasing, especially in these novel habitats like what we have in Alberta. So to fill these knowledge gaps, we essentially need to improve monitoring. And that's a big knowledge gap, is where mountain pine beetles are. And when you look at old other bark beetle, post-bark beetle outbreak assessments or strategic assessments of bark beetles, they always highlight the need for improved monitoring. And that's because we use fixed wing planes or helicopters, which are expensive and have safety issues, and also have just some data issues associated with them. Or we use trapping, which again, our traps are made for usually higher populations. That's where they were tested, and they only have a limited range around that trap. So knowing where beetles are could improve our prevention of the eastern spread of mountain pine beetle from Alberta into Saskatchewan. So that's where some work I did with in U of A with Nadir Erbligan. We actually went out and improved pheromone formulations to kind of make it specific for Alberta. And we did that by using the synthetic pheromones that the province use and companies use to attract mountain pine beetle. And we essentially changed the formulation so that we can make it suitable for our low densities and our novel habitats here in Alberta. And in doing that, we found that adding not just the commercial pheromones, so the chemicals that beetles make, but also the two chemicals that the tree makes, the terpinolene and myrcene. Normally, when the province goes and buys aggregation pheromones to put in traps for monitoring, those lures only have either terpinolene or myrcene plus the pheromone. But we found when you added them both, that you got increased catches, and particularly at low populations. And we also looked at optimal baiting formations and distances between stations to essentially help the province optimize their detection strategy for mountain pine beetle. And in doing this work, so we improved the pheromone by adding both the chemicals, tree chemicals, to that lure. We also wanted to understand whether there was evidence of density dependence of mountain pine beetle to its own pheromone. And so we kind of got this idea because in the literature, in BC, a researcher found at high populations of mountain pine beetle, if you increased the amount of this beetle-produced pheromone, it's a traction pheromone called transverbenol, that you would continue to get more and more beetles caught in your trap. However, at low populations, in BC lodgepole pine, if you increase the amount of this beetle-produced pheromone, that wasn't a linear relationship, and actually at really high amounts of the pheromone, there was a decrease in trap catches. So we wanted to investigate that more in Alberta. And so we set out to look at whether attraction varies with this local population of density. Again, trying to find the best lure that the province and companies can use to monitor mountain pine beetle while they're in this novel Alberta habitat and at low populations. So we kind of did this factorial design where we altered the amount of the beetle-produced pheromone, the transverbenol, and the amount of tree chemicals, so myrcene and terpinolene. And this full kind of factorial design where we wanted to test all of them in all combinations. And we did this from 2017 to 19. And we found that, yes, if you look at this ANOVA table, that there was a significant effect, a significant interaction in the lures and the local population. We estimated this local population using the aerial detection survey. And so the lures performed different in that it caught different amount of beetles depending on that local population that was indicated by the aerial detection survey. And we wanted to, again, do this, but pick out the two lures that we could use to detect beetles in low and high populations while they're in Alberta. So we picked out the two most different, which were the standard beetle pheromone lures that you would get that you could buy, the province buys, and then we tested that against adding double the transverbenol, which again is that beetle-produced pheromone. And we found that yes, they performed differently in that they caught different numbers of beetles depending on that local population. And this was only in 2 sites that we tested this in, and these were all in Lodgepole, probably it's in the hybrid zone north of Whitecourt. And so we found that when you are in a high population situation, high relative to Alberta standards, that, or at least the eastern edge of Alberta standards, we had higher catches when you used the standard pheromone, but when you were at lower populations, like very low, we found that increasing the transverbenol, so the amount of pheromone that the beetles make, increased our catches. So in a low population situation, beetles could be more sensitive to their own pheromone to try to find trees that are already attacked because they use that to overwhelm the defenses in the tree. So they are, at low populations, they're very sensitive to their transverbenol, their own aggregation pheromone. And so what future we want in the future, we are wanting to expand this and do a more extensive survey to see whether this this phenomena is something that we could use to essentially not just detect low populations, but also be able to identify if populations are increasing or decreasing. So in an experiment that me and two researchers at the CFS are going to do this summer in British Columbia and Alberta is we're going to put out some series of transverbenal and other lures that we want to test and see if we can not just locate low populations, but see if we can find if there's a density dependent response to their own pheromone, with the idea that at very low populations in this population graph, we would find more sensitivity to high transverbenal, but when they are starting to transition, that we would see transition to outbreak, that we would see this lower sensitivity to transverbenal. And that could be used as an indicator by managers to that beetle populations are increasing. And finally, I want to talk about Beauveria. So it's a biocontrol agent. This is work that was done in University of Alberta that was I'm just going to present the lab portion, but there was ongoing field portion to it also. But so Beauveria, what is it? It's an entomopathogenic fungus. So it's a fungus that kills beetles. You can see in that photo, the white fuzz is the mycelium. That's the fungus growing on those dead mountain pine beetle. And it's used as a biocontrol agent in agricultural system. It's kind of a generalist fungi, so it can infect a large diversity of insects, but you can find strains of it that just infect a narrow range of insects. And as you can see, they are dendroctonous species, so mountain pine beetle and other bark beetles that are in that genus are susceptible to Beauveria. And an interesting part that goes back to the chemical aspect of how trees and beetles interact is strains can be really sensitive to the environment. They can be sensitive to UV, but importantly to the chemistry of the tree. So this may impact how we use it as if we do use it as a biocontrol agent. So in some recent lab tests and some ongoing field tests, they found out that they identified that there is different virulence, so the ability to kill mountain pine beetle depending on the strain. So we could use that to identify strains that are kill mountain pine beetle the fastest. And we found that we tested whether they tolerated UV and lodgepole pine and jack pine monoterpenes, plus the mountain pine beetle pheromone transverbenal. So in this work that Albert Rosana did in his PhD, he found that faster mortality happened than controls when those beetles were infected with Beauveria bassiana, with this biocontrol. And he essentially found effective, he also identified the effective dose to kill, which could be then used to formulate a deployment strategy. He also found that strains of Beauveria tolerated lodgepole pine monoterpenes, so that profile of monoterpenes that I first showed you in that heat map, better than the profile of monoterpenes than that jack pine had. So that could be a consideration when whether this would be a viable biocontrol when putting in maybe a jack pine situation, or the strains we might have to find strains that are less sensitive to the jack pine monoterpenes. And finally, he found that strains varied in their tolerance to UV. So, but importantly, he could essentially improve UV tolerance using generational populations exposed to increasing times of UV. And this is really important because when you go to deploy this, you need to, say, spray Beauveria bassiana on a tree, or put Beauveria bassiana into a trap that traps beetles that then go out and disseminate. They're going to be in contact with UV, and if UV kills the fungus, then it's not a very effective biocontrol. So future work needs to be done. A lot of future work needs to be done on the effectiveness of Beauveria bassiana in the field. So we need to test the efficacy actually on trees in the field. We also need to develop some deployment formulations and methods. So one method could be to spray the tree with Beauveria bassiana and have it stick to it. So when beetles attack, they get spores on them that eventually infect and kill them. Or another way could be to deploy Beauveria with a drone, but then you have the canopy issue. Like there's multiple issues that need to be dealt with, and so the formulation may vary based on that deployment. Also, we need to identify the effectiveness of this at relevant spatial and temporal scales. So Albert found that Beauveria bassiana killed beetles maybe 1 or 2 days earlier than the control. Is that effective when you put that into the field? Is that enough to reduce populations? And also address non-targets. So this, for the control for the lab bioassays, honeybees were used as a control. But as I said earlier, Beauveria bassiana tends to have a wide range of hosts that it kills. So even though it did not kill the strains that we used in this experiment on mountain pine beetle, in that it didn't kill the honeybees doesn't mean it won't kill many of the native species of beetles or other insects that we have here. So we need to understand non-target effects and how to minimize that. So kind of in summary, we need to expect that mountain pine beetle is now part of our forests in Alberta. Stand suitability models still indicate the continuation of suitability of our boreal forests, even though we are at these really low populations right now, and we had some cold winters. We may expect mountain pine beetle to just be part of our forest now. But we need to understand these filling in the holes of uncertainty that we have, such as how beetles perform in jack pine, how beetles perform in the different chemicals that the different chemistries that the trees have here, and also what biocontrol or management that we can use here. We also need to take advantage of these low populations in the expanded range to control them. So we need to understand what maintains these low populations and what forests are actually accessible to them, because our forest structure, especially in jack pine, is different from that of lodgepole pine. So how does that impact how mountain pine beetle may access those jack pine forests? And we also need to improve our monitoring tools, particularly to for low populations, because it's very expensive and there's safety concerns with aerial detection using planes or helicopters. And at low populations, it's not a very effective means anyway. So how do we detect that. So one way that I'm going to try to look at that is to understand this density dependent behavior, and maybe some other ways that we can is look for suitable trees using, say, drones, or some other means that we can identify what suitable population is out there, and particularly before they erupt. And finally, investigating control measures. And one possibility is Beauveria bassiana. It hasn't been used in mountain pine beetle. It's been investigated in other bark beetles with varying success, not very much success in the field. So a lot of work needs to be done there, but maybe this is an opportunity in Alberta to develop that. So I'd like to thank many of the people I worked with, Nadir and Barb at U of A, and my colleagues here at CFS, plus grad students that worked on this, and the amazing summer assistants and student assistants that worked on this, plus funders for the two projects, the Res4 project and the trapping project. And with that, if there are any questions, I'd love to answer them. And I think I'll stop sharing my screen now. What impact do our increased forest fires have? Yeah, that's a great question. That's something that I'm going to be looking at a little bit, but more in the salvage logging aspect soon. So, bark beetles, so essentially that's removing host trees from, or suitable host trees, or the amount of host trees from the beetle population's ability to use. However, we still aren't at the fire regime, we're still dealing with the kind of a fire suppression legacy right now. And so we still have a lot of large diameter trees, because that's what mountain pine beetle requires on our landscape, because even though we've had some of these large fires, they still haven't broken up our landscape to make it more heterogeneous, so beetle populations can't blow up. So the large fires probably aren't doing much from a beetle population perspective, other than beetle proofing where that fire was for a few years, for many years after that. I know the province has been, or the parks have been using controlled burns as a way to essentially remove hosts from mountain pine beetles' ability to infest it. And that has moderately worked, I guess, in the parks. At least it is dealing with the problem. It's introducing fire back into our landscapes, and it does reduce the amount of hosts in the landscape. And some recent research looked at that these fire damaged trees don't actually increase the beetle population, because that's true for many other bark beetles. They take advantage of fire damaged trees to build up populations, but that really wasn't the case in the prescribed fires that were being done. And I guess one was a wildfire in the parks in Alberta. So that was a good thing, that along the edges where you have your beetle, or where you have your fire damaged trees, it doesn't increase beetle populations.
What do you think the most challenging part of introducing biocontrols is?
I think there's two things that really bother me. One is from a biologist perspective. I don't like the non-target effects that Beauveria bassiana will have. So even though that experiment used another insect as like a control, so we have plenty of beetles out here. Can we find a strain that is just going to impact mountain pine beetle, or maybe just Dendroctonus, or I really don't know. And so the non-target effects is what I would have a large issue with, because it's all the other insects that live in the forest that actually help control mountain pine beetle. So we don't want to kill those while also trying to kill mountain pine beetle. And another one is the formulation and the method of deployment. So others have looked at this in spruce beetle, in North American spruce beetle, but also European spruce beetle, and they found they were just unsuccessful in their attempts to put Beauveria bassiana on a tree to effectively kill the beetles that were attacking it. And that doesn't bode well that we're going to find, or that Albert is going to find the best way, and a way that will work here in Alberta in mountain pine beetle. But yeah, it means it's going to be very challenging finding the best way to put Beauveria out there to be effective against mountain pine beetle. But in my heart, I'm more worried about the non-targets.
Well, thank you. If we don't have any other questions, oh, there's another question.
Oh, there it is.
Are the traps effective at attracting only mountain pine beetle, or is there bycatch as well?
Yeah, there are bycatch, and that could be slightly an issue, but the bycatch that we get are usually things that just kind of fell in that you would get if you were just to put a trap out there without lures. So we get randomly a lot of sawfly larvae. I don't know, they're just dropping in. Like, they don't got wings and they're not climbing in. So I don't know how they're getting in there. And we tend not to get many of the predators and competitors that, again, control mountain pine beetle when mountain pine beetles at low populations. These tend not to be very sensitive to beetle pheromones, but there are some bycatch when doing the traps. But it's nothing like what would happen with Beauveria probably. But yeah, there is bycatches. If anyone has any further questions, you can contact me. My email is just my name at nrcan-rncan.gc.ca. You can look me up on the internet. So if you have any more questions or anything, please email me. I'd be very interested in talking with people.